All right. Wasn't sure how many more people were going to come in, but I do want to start by talking about the midterm. I just didn't want to have to repeat myself. So I was in the process of writing up an announcement this morning, just letting you guys know that I released the midterm grades. So you have access to those on Moodle right now. You actually have access to the full exam, so you can go in and look at your attempt, all the questions, and the right answers. Usually I would do sort of a standard book a room and have in-person midterm viewings to try and keep questions private. But because of the online setup, it's so much easier to just do it this way. And then you guys also get those resources to do as well. So I figured that was sort of the best approach we could have gone with. Um, that being said, uh, I've talked to a couple of students, and so I've taught quite a few sections of intro, enough that I have a predicted sort of distribution of grades. I have a predicted timeline for stuff, and I know what average I would expect. And anytime I get deviation from that, I know that something's gone on. So in this case, I found that the class average was a little bit lower than I would have liked, which I'm going to attribute to the fact that we got stuck with an online last minute assignment or exam that was so different from what we had prepared for in class. And I know that that absolutely has to affect people's grades. So, since my goal is to actually get a representative measure of ability, and something like this can disrupt that, I'm always willing to make adjustments and corrections, especially where we're back to in person now, and we should be back to our standard way of writing exams from here on out. So to that effect, I am going to be making two adjustments. Again, I'm going to be posting this on Moodle so you can actually see all the details written out. I just left early because I wasn't sure how long it would take to get in the front door, so I didn't get a chance to finish. But the first thing that I wanted to do was a straight correction to the class average, which is basically I took the best part of the class and I divided everyone's score by that instead of the actual total number of questions. Usually I would just remove questions, like if there was a question that everyone didn't get, I'd pull that out. But we didn't see that. There wasn't sort of a systematic problem. It was sort of an everywhere overall, which made me consider that it was the method of delivery. So I have on here that it's out of 43. I don't think that's accurate. That was sort of a ballpark guess for trying to put stuff together. So I'll be dividing by somewhere in the range of 43, probably a little bit smaller than that. And basically all that does is it's going to be a straight boost to your grade. So I did some quick math at the bottom where it says if you originally had 30 out of 45, that was a 67. If it's 30 out of 43, it's now a 70. So I'll give you the exact numbers in the announcement once I work it out. It's all worked out in a spreadsheet, not on this laptop. It's just been one of those days. So that's the first quick and easy thing. And um, with that one, it was kind of hard because I guess the other thing that gave me that hint that it was something with the delivery method, I had some students who did great, some students who got almost perfect in the class, and then some who just struggled with what I assume is the delivery. So that's where the second method comes in, where I'm going to add in a second grading scheme. Basically, I'm going to calculate your grade two ways. One, the exact standard that was set up in our syllabus. So the midterms are each worth 30 or 25%, final is worth 30. I'm going to do an alternative one. I'll calculate this as well, where I'll reduce the weight of the first midterm. I'll drop it down to only 15% and move the extra 10% to the second midterm and the final. And then that way, the first midterm didn't go so well for you. It isn't going to drag your mark down. So I'm going to calculate both and I'll just see what's the best option. So if you did really great on the first midterm, then that's fine. That can still count as your 25%. I'll calculate both and take the best. And like I said, I'm going to put this info up on our Moodle so that you have access to that. Um, I don't have a way to tell Moodle that our score is out of 43 or whatever it ends up being, because if I rescale the grades, it just takes a proportion. So it adjusts it so the percent is the same, which defeats our purpose. That's why the announcement is going to tell you what it's out of, and I manually calculate the grade myself, just so that you don't panic when you see it out of 45. But yeah, so hopefully those two corrections make sense. As I said, I've done this enough that I know where I want to be, and then when we have extreme events like this, it doesn't always go to plan. So we're just making a couple of little adjustments on the fly. But I will only ever take the better of the options from these. I won't ever backwards. I'll only sort of scoot it the other way. Because if you did really well, then that's a good thing. But if something's gone wrong, that's usually a systematic issue. So hopefully that makes sense. I like to be upfront and transparent about stuff like this because it's it really matters to you guys. So this is how it's set up and why. Um, if you have any other questions about that, or if you think of anything, you can always send me an email or chat after class. But yeah, you have access to all of those questions. You can look stuff up on Moodle and go through your exam at your leisure now. And I'll leave you with access for that for the rest of the semester. Um, I had another thing that I wanted to talk about. What was it? No, I forgot it in my other class too. I remembered in the other one, but I'm not going to remember here. Oh, nope, there it goes. Okay, so I have my whole recording set up here again. This is what I did at home. This is what I'm doing here. So I'm still going to be recording and posting everything on our Moodle. So if you find it a hassle to get in now, if you just want to stay home in bed one day, no worries. I'm going to be recording and posting. I actually upload in between my classes, so I do 3 classes back to back. And while I'm running through the halls, it's processing on YouTube. So you should have access to everything pretty much immediately if ever you aren't comfortable being here. Okay, those were the things I wanted to hit. So now we're going to get into actual content after a long preamble, but we're going to start talking about some of our uh, smaller in scope, not necessarily in importance, of our senses. And as I mentioned in our recordings, I don't cover this chapter in the same order that the textbook covers it. I find that every single chapter launches right into vision, throws you in at the deep end, and hopes you figure it out. And I like to actually build up to vision. I like to start with our auditory system. Well, actually, I start with some of the basic terms and concepts, then go into our sense of hearing, and then sort of cover all the stuff in between. And then we wrap up with vision. So that's the logic behind that pathway here. But we're going to start here with our two chemical senses, which are taste and smell. And these two are fairly closely intertwined. We'll talk about how they interact with one another towards the end of this part. And then we'll talk about our sense of touch and then our two bodily senses, our kinesthetic and vestibular systems. And you're going to notice right off the bat that each of these systems, because I've just reeled off 5 different systems. We're going to cover them all in one class. So clearly each of these systems is covered in a lot less detail than either our auditory system or our visual system. And that's intentional. The first reason, because we only have a set amount of time. The second reason is a lot of these senses, we still don't know exactly how they work. As much as it drives me nuts, there's a lot of stuff where we'll talk about this is how we think it works, or this is how it might work, or this is what research says might be going on. We don't actually have solid answers for all of these. So you'll find we leave it at theoretical in a lot of places. So that's our rationale here as well. But starting with our sense of smell or olfaction, we're going to do that standard thing where we talk about what are our stimuli, what are our receptors, and how do they connect. So our physical stimuli for our sense of smell are odorants or chemical substances that are carried in the air. The absolute coolest description I've ever seen in a textbook was to call them volatile chemical compounds, which from the scientific perspective on each of those words is accurate. Volatile just means that they aerosolize or evaporate at room temperature. Chemical because everything is chemicals, and compounds because it's part of the descriptor. So it makes sense. It's just a very, I don't know, intense description to include. But for our sense of smell, it's basically stuff that's floating around in the air and is capable of dissolving in the mucus lining within our sort of sinuses. And we'll find that that's very similar to our chemical sense of taste, where it's the chemicals in our food that dissolves in our saliva to then trigger receptors. And here, our olfactory receptors are called olfactory cilia. And they always have some iteration of this exact image in every psychology textbook. None of them seem overly helpful at first glance, and they're all equally weird. But the way that I describe it to people is if you've ever had a sinus infection or that feeling sort of at the back of your mouth, behind your nose, like inside your skull, it gets itchy and tingly when you know you're getting sick, that's where your sinuses are. And so below our skull, in our heads, or sorry, below our brain, in our heads, we have sort of these open passages lined in mucus. So lots of open sinus areas. And one of them connects directly to our nasal passages, right to our nose. And that's where those smells that we breathe in are coming in and sticking to the mucus lining and triggering those cilia or olfactory cilia. So these are our receptors. So they're shown here as kind of a blue spider looking things. They're reaching out. And so when our dissolved chemicals bind with those receptors, it's going to send a signal back into the olfactory bulb, which is at the base of our brain. It's really neat. If I show you the pathway written out here, we don't actually go through the thalamus because that olfactory bulb is right touching on the bottom of our brain. It can send its signal directly into the brain without having to be rerouted through the thalamus. And pretty much every other sense that we talk about goes through the thalamus, distributed to the right part of the brain that way. But our sense of smell doesn't. It's those chemicals bind at those olfactory cilia. There's transduction, which is going to turn that sensation into some kind of neural impulse that can be passed along, hits the olfactory nerve and into that bulb at the base of our brain, but no thalamus. And so the theory here is that our sense of smell evolved so long ago. It's such a basal ability that it happened before we had complex structures in our brain like the thalamus. So our sense of smell would have evolved very, very early on, and then everything else has sort of evolved since. That's at least the going theory that they have. Actually, before I move on, I will talk about the fact that humans overall, we tend to have a very poor sense of smell. And it's not because we can't smell all of the smells that other species can, it's because we're not sensitive to smells. So we need a much higher concentration of an odor to be able to smell something. So if we had a bloodhound in here, they could have a couple of particles and they'd figure out the scent and be perfectly fine. But for us, we need, I think it's somewhere in the range of like 40,000 times the concentration to be able to smell the same thing they can. So humans have a very poor sense of smell. We are very insensate when it comes to smells. And I always kind of make the joke that I'm on the bad end of the spectrum where my sense of smell is absolutely atrocious and has been for a really long time. Like I don't remember ever being able to smell. So I have like 5 or 6 scents that I can kind of tell apart, but mostly it's there's a smell and it's intense and I'm not sure if it's good or bad, but there is a smell, which has led to some interesting conversations when I mislabel things. But when we start talking about taste and how close smell and taste get involved with one another, you'll realize why that has been somewhat problematic in the last year and a half. But we can also talk a little bit about sensory adaptation. So, like most of the other senses that we've talked about and that we will talk about, our sense of smell shows sensory adaptation. I loved when Febreze came out with their nose blind commercials because that is the best example of sensory adaptation for smell I have ever seen. So if you haven't watched them, they're really short clips. You can find them on YouTube, and I find them hilarious. But it's basically the idea that maybe you drive around in your car for a while and it doesn't smell bad at all, but you go and pick someone else up and they get into your car and it stinks like dog. You don't notice it because you've gone nose blind to it. which means you've adapted to that smell. But someone new who's just coming into that smell is going to be overwhelmed by it. And so the way that it works is our perceived strength of an odor usually fades to half or even less of its original strength in as little as 4 minutes. So when you walk into a bakery and it smells like fresh baked bread and everything's amazing, it's good for a couple minutes, and then you stop noticing it. And that's because a lot of our senses, a lot of the reason for our sensory adaptation is we only care about stuff that's changing. So when we're in a situation and it always smells like pine trees, well, that's no big deal. You live in a pine forest, we don't care. But if one day it smells different, now it smells like bear, that's important because it means there's probably now a bear. So the stuff that's constant we can block out, but new and changing sensations should always be brought to our attention. So that's a little bit of background on why adaptation works. Um, and then there's a lot of really weird studies about our sense of smell. Um, this is, again, beyond the scope of the course as per usual, but I find it strange. The one study that I've read about senses of smell is they had three groups. So a group of university undergraduate males who they had put on white cotton t-shirts and go play basketball for an hour. They also took a picture of each of those participants and then they matched sort of like, okay, so this picture is this individual and that shirt is this individual. Great. Then they had a smelling group and a visual group. So one group would smell all the t-shirts and rate them on attractiveness. And another group would look at their pictures and rate them on attractiveness. And they looked at whether the scores matched. And they've done this in different populations, different groups, multiple times. There's always a really strong correlation between the smell rating and the attractive rating from pictures, which is super gross and super weird, but also one of those things that seems to happen all the time. So that's sort of tied into the idea of pheromones and hidden chemical messages that we can perceive but aren't aware of. But I always like talking about that study because it's neat, but it's also really um, and across multiple odor tasks, not just sniffing t-shirts. We tend to find that females tend to do better than males in humans. So they're a lot better at odor recognition tasks. So for those kinds of studies, I would definitely be an outlier for them. But if we continue on into our sense of taste, we have again our physical stimulus, soluble chemical substances. So this time, these are substances that can dissolve in our saliva. And our receptor cells are found within taste buds. And this is one of my favorite things about this chapter is when you think of taste buds, you think of those little bumps on your tongue. But in fact, taste buds are the valleys between those bumps, which blows some people's minds. But if we look at this kind of outdated image here, we see that the taste buds are labeled as the little valleys, the grooves in between those bumps. So the bumps themselves aren't taste buds, they're just bumps. And then our taste buds, if we can see here, I remember which way the mouse goes, so that would be the valley in between the bumps, and then we have a collection of cells. So each taste bud has 50 to 100 different taste sensitive cells that will then pass on that information, which is really cool. But we have a couple of different types of taste receptors within those taste buds. And this is where we get into that not everyone agrees category. You will see general agreement that there are four tastes: sweet, salty, sour, and bitter. There's now a lot of evidence for a fifth taste, which is umami, sort of a savory flavor. Um, you won't find it on pretty much any of the graphics in textbooks though, because that's come out in the last sort of 10 or so years, maybe less, and not everyone agrees on how we should interpret it. So unfortunately, most of our data is just looking at the four main flavors. But in the next couple of years, we're going to start seeing that work its way into the literature quite a bit more. Another of those misconceptions that we tend to have, usually because you are taught this in grade school, is that sort of taste map where you can only taste bitter at the back and only taste sweet and then sweet or salty and sour are on the sides. And that's a little bit misleading because we have all of our different kinds of taste receptors all across our tongue, but we have slightly different concentrations in different locations. We can look at, again, a slightly outdated image here, but we have those bar graphs where each color is a different kind of receptor. So dark blue is for bitter, and we see that at the back of the tongue, yes, there are a lot of bitter receptors there, but there are other types of receptors too. So that misconception that there are only certain places that certain flavors can be perceived isn't accurate. We have different distributions, different concentrations of all of these different receptors in those different places. So it's a little bit more complex than that. And then for this image, we talked about how that's your taste bud at the bottom. And it's mostly here to show you that those receptors are going to feed back into our nerves to send that information into our brain. So for our pathway, we have our taste buds. They convert that information into a neural impulse. They transduce the signal into something that can be passed along our neurons. It's going to head through our thalamus and into the cortex. So our thalamus is going to route that information to the parts of the brain where it matters. And we can talk just a little bit about our preference for tastes here. So again, there's a lot more research to be done on this, but some of our basic taste preferences seem to be innate. And there's some pretty good agreement across multiple studies and lots of evidence that goes along with this. But the easiest of these to sort of visualize is the idea that when you're very young, you tend to like sweet things. Children love sweet foods. And if we think about it sort of evolutionarily, a lot of sweet foods tend to also be very high in calories. And we have to sort of separate ourselves from society right now, where we have processed sugars and easy access to lots of different things. But if we were looking at living in nature and you had access to things like berries, where there's lots of resources, lots of nutrients that would be good in those foods. And so we have strong flavors in some very high calorie or calorie dense foods. And when you're a child, the important thing is to get as many nutrients as you can to grow and get stronger and be bigger and stronger and healthier. So it makes sense that we're hardwired to seek out what should be high calorie foods at that age. And we do see that preference where children love sweet things. And to make us even more certain of this, as we get older, we tend to decrease our preference for sweet things. I thought that this would never ever happen to me because I have had a sweet forever. And when I was younger, if you tried to give me dark chocolate, it was the worst. It never happened. But towards my sort of mid 20s, I started being able to tolerate dark chocolate. And now sometimes there are days when I would rather a dark chocolate bar than a milk chocolate, even though that seems absolutely crazy. But apparently, it's some of our biological developmental processes, and it seems to be consistent for most people. Um, we also have some taste preferences that are learned. So some of the basic stuff is hardwired, but most of the rest of it's going to be learned. So whatever we're exposed to. And of course, because it's a psychology class, we want to think about other things like associations. So if we think about maybe there's a meal and it's maybe not your favorite meal, but your family always eats it on a particular holiday. So whenever you get that food, it makes you think of that holiday and eating with those people. So all of a sudden you like that food, not because of the food, but because of what you associate it with. There's also stuff like preferences from just what you're exposed to. So my family was from Nova Scotia. We eat a lot of boiled things, boiled fish, boiled potatoes. It was very bland and boring. But for me, like a comfort meal would be something like boiled potatoes and cod, and that would be fine. But for most people, that would be highly unappealing. Maybe you don't love that much salt all at once, but whatever you're used to, right? So preferences can be learned, and there's a lot more to it than just how you taste things. Another thing that we could consider in our ability to taste and what we end up liking is that not everyone has the same ability to taste. So some people are sort of middle of the road, they're average, which most people should be. But we also have people who taste very, very well. They have a strong sense of taste. Those would be super tasters. Or you can have people like myself who have almost no sense of taste. We are non tasters. And people who tend to be super tasters, you have a friend who can eat a meal and tell you every spice that's in that dish. That's a super taster. I have one of those and I feel ridiculous because they'll tell me the list of spices. I'm like, it's spicy or sweet or like something generic. But that or that ability to perceive can influence preference. People who can tell all of those spices, all of those flavors coming together, they're a lot more likely to eat healthy because they can tell exactly what's in their food. They're also more likely to make their own food because they'd rather have control over each and every ingredient going into it. Or you could be like me and just be fine with craft dinner anytime. But this is where we get into our combination of taste and smell, where we get into the idea of flavor. So we combine all of those senses, our taste and our smell, in the orbital frontal cortex brain. And our flavor is a combination of taste and smell, as well as a little bit of that tactile sensation of food in your mouth. So if you think of an apple, it's not just that flavor, it's not just that smell, it's also that crunchiness, which is very distinct and different from, say, a cooked apple if you were in an apple pie. And so all of these combinations really come together. So if you think of a time that you had a cold, a really bad head cold, you're really stuffy, your nose is all stuffed up, and you go to eat a meal and it tastes boring and bland, not because the meal is different at all, but because you can't smell it. And that's why, for me, having very little sense of smell leads to a low sense of taste, which again, problematic the last couple of years. But when you grew up with it, that's what you're used to. For everyone else, you kind of get intermittent experiences of that when you have a head cold. So if you lose that sense of smell, a lot of taste goes with it because we incorporate those scents together and sort of put them together as flavor. All right. So, as I said, these are each going to be fairly short. And that was one of the longer ones we're talking about today. So, next, we're going to talk about our sense of touch. We're going to wrap up the shortest two. But for our sense of touch, we're pretty much talking about our skin and the receptors that are in our skin. And this is another one of those where it's a lot more complicated than we can talk about, but the general agreement is that human skin has at least 6 types of sensory receptors, uh, potentially a lot more, but I can't find the upper limit on what they'll agree on. So they seem to agree on at least 6. We're going to talk about 4, but only 2. So we're going to talk about sort of a tactile perception of touch on our skin. We're going to talk about temperature. We have warm and cold sensors, as well as pain. And so with these, all of our physical stimuli are going to be different kinds of mechanical, thermal, or chemical energy that's in contact with our skin. So for thermal energy, things like temperature, if you know about what heat is, it's higher energy, things vibrating quickly. So we would have sensors that pick up that extra thermal energy. Or cold would be a loss of that energy, less vibrations. So we could measure cold that way. So that's sort of the stuff that those temperature sensors. Mechanical sensation, that's just basically touching your hand. You know exactly where you are poking because you have stimulated those receptors in your skin. And then you can have chemical energy. So if you've ever had like an icy hot rub, if you've pulled a muscle or something and it feels hot and cold and both, that's chemical energy causing some different reactions as well. So lots of different sensors, and we can have much more than these, but we'll keep it to our basic. And so for our basic pathway here, we have our sensory receptors, and I forgot to put in the fact that there is going to be transduction. There is going to be that passing on of some kind of neural impulse that will come after the sensory receptors, because that's what the sensory receptors do. They take that input of whatever they've been touched with or interacted with, and they turn it into a transmittable neural impulse. So that would happen here. And then it would send it on to the spinal column, our brain stem. After the brain stem, there's a crossover point. So when we talked about the different parts of the brain and how our brain interacts with the rest of our body, we mentioned the fact that the right side of our brain deals with the left side of our body, and the left side of the brain deals with the right side of the body. So there's a crossover there, so that sensation coming in from the left side will travel up to the right hemisphere and vice versa. So that's why that's happening there. And this routes through our thalamus, we already discussed, and it's going to land in our somatosensory cortex in our parietal lobe. So those are those strips along the top of your head. All right, so let's start by talking about the pressure type sensor. So that's that tactile touching on your skin sensation. So we have specific cells, specific areas that are sensitive and paying attention to specific patches of our skin. I found the best way to talk about this is to think of like a bullseye. At the center is the point where we are making contact. That is the most stimulated area. And that center is going to send a message to our brain to say, right here, I'm feeling something. So we get an excitatory signal from the middle saying this is where the stimulation is coming from. And so our skin, those patches or areas of skin, are connected to different neurons that are going to send signals up. So in the middle, we have that one spot, that target point that's being triggered, and the area around it is being also slightly impacted. Because when something touches your skin, it doesn't affect just that exact part of your skin. There's sort of an indirect interaction around it. So those areas around it are going to have inhibitory signals. So instead of increased neuronal firing, we're going to have decreased firing. So that helps our brain target exactly where that sensation is being perceived. So right in the middle of the bullseye, that's where we're being touched. And the area around it is where it is not. So we have that excitatory center and inhibitory surrounding. And our brain is able to interpret which specific pathways, which specific connections of neurons are leading to or sort of activated when different parts of our skin are touched. So we have sort of a predictable arrangement. And if you remember back to our discussion on the somatosensory cortex, different parts of our body will have sort of larger or smaller stretches in our brain based on how sensitive they are. So things like our hands are very, very tactilely sensitive. They make up a very large portion of that somatosensory cortex. So that's because the parts of our sort of the skin here would be divided up and fed back more neurons than somewhere where we're less sensitive, like say on our torso. And I found a really good example for this. I don't know if everyone did this as kids, but sometimes you'd sit around and you'd have someone draw circles up your arm and try and find the midpoint in your arm and how close could they get with your elbow. And there's a patch in here where for about 10 or 15 centimeters, it's really hard to tell exactly where they are. And so you can find that you're off by 5 or 10 centimeters. You swore they were exactly at the end, at the inside of your elbow. And that's just because those patches of skin are much larger. So a larger area is being fed through just one neural pathway instead of multiple, like you would have had on your fingers. So less accuracy just means that we're looking at a larger surface area that's being fed by the same number of neurons. So that gives us this really fantastic ability to localize sensation. phenomenal at this. We can also talk about that idea of sensory adaptation. So this is when you feel things on your skin. So again, stuff that's constant, there's no point in paying attention to it. So you put your shirt on in the morning. Maybe it's a little bit itchy when you get started. But by the time you're ready to leave the house, you're used to it and you don't think about it again until your instructor mentions it, and now you're very uncomfortably aware of it. But that's how sensory adaptation works. So if it's constant, you can ignore it. But if there was a fly in the room and it landed on the side of your neck, you would immediately be aware of that because it's activating that tactile feeling on your skin and it's new, it's different, it's changing, so it matters. So your attention is drawn to that, but you wouldn't necessarily care about how your clothes feel unless you're made to think about it. And so the next one we can talk about are our temperature and pain sensors. And we're going to talk about temperature very vaguely. I've already mentioned the sort of hot versus cold, high versus low energy, but both temperature and pain receptors are free nerve endings. What this means is that there are no specialized receptor cells. They don't have the olfactory cilia. They don't have rods and cones like we do in our eyes or hair cells like we do in our ears. For temperature and pain, our neurons are just what is there, what's feeling that sensation. So the dendrites of our neurons that are in our skin are what ends up being stimulated to send that neural impulse into the brain. So these aren't specialized cells, these are just neurons or free nerve endings, as they talk about here, because when you're outside of the central nervous system, we go with nerves. And with pain, because like I said, we're going to focus on pain more than temperature, pain has two distinct pathways, a fast pathway and a slow pathway. And I actually have a visual here, not that you need to know the visual, I just find it a little bit easier to look at. So the red pathway, that A delta fiber, so this red one here is a much faster pathway. So it's going to go straight to our somatosensory cortex. And the nerves or the neurons that make up that A delta fiber pathway are all myelinated. And if we remember, myelin helps those signals travel faster because you can hop between the nodes of Ranvier, you get a much faster transmission of those signals. For C fiber pathway, those neurons are unmyelinated. So they're going to travel much more slowly. And they're going to get in here and they're going to end up sort of generally distributed to the rest of the brain. It doesn't make an immediate line straight for our somatosensory cortex. So the signal travels slower and it's going to be less immediately direct. And the layout here is less important. I just like showing that there are differences there. So for our fast pathway, those are going to give us a quick, within a fraction of a second, and accurate localization of where pain is coming from. So it's going to happen right off the bat and it's going to tell us exactly where it hurts. Our slow pathway is going to be less localized, slower, but also longer lasting. So that's sort of an aching or burning feeling that persists. And the best way to think of this is if you've ever kicked your coffee table. You know that feeling where immediately right off the bat, there's sort of a sharp intake of breath that really hurts, and you have just long enough to go, oh no, this is going to hurt. And then you get that wash of like your whole foot hurts. That first feeling, that instant, oh no, I stubbed my toe, is that fast pathway. And that later, slower realization of big pain, that's the slow pathway. So they're both working together. They're both telling us that we're hurt, but one is quick and goes away and tells us exactly where. And the other one is slower. When it shows up, it's going to stick around for a long time and it's a lot generaler or more general. So those are the two pathways there. All right, and of course, if we're going to talk about pain, we need to talk about perception of pain. This is a psychology class after all. And this is one of those really neat topics that I could go on forever, but we don't have the time for. But our perception of pain is influenced by things like our expectations, our personality, our current mood, all sorts of different factors. So this ties back into the fact that everyone has sort of their own subjective way of interpreting things. Pain absolutely fits that rule for psychology. So if you look at something that makes you happy, it tends to reduce your perception of pain. Maybe you stub your toe and then look at cute pictures of kittens, and that will help. The intention behind pain can actually change your perception. So if you're told that someone intentionally caused you pain versus if it was an accidental pain, that's going to matter. The intentional pain hurts a lot more than the unintentional. This also works for stuff like with depression. When you have a low overall mood, everything feels worse. Pain is more acute. And the problem with depression is that that pain can then feed back and cause more depressed feelings, which causes more feelings of pain, and it becomes a vicious cycle. One of the reasons why it's so hard to break free of. We can look at things like expectations in the placebo effect, where if you give someone a sugar pill and tell them that it's a painkiller, their perception of pain will decrease. They'll assume that it's helping, and then it does. And there's lots of research on cultural differences in pain perception. And that's one of those areas that, again, we have to look at the research with a little bit of a critical eye. And that's because a lot of earlier research was looking for either biological differences or hardwired differences between people from different places, which is almost always problematic. But more recent research has shown that a lot of what happens in the differences we see between cultures isn't necessarily in their perception of pain, but in the way that they frame pain and interpret it. So one of the basic examples that I can get across quickly is we tend to group cultures into being either individualistic or collectivistic. So North America would be a primarily individualistic culture. You focus on yourself. Everything is about what you are accomplishing and what you are doing. Other cultures might be collectivistic. So I believe Japan falls under that category, where they focus on the community and success for you is a success for your group. And so we could look at things like, say, the effect of the pandemic and isolation. If you're only meant, or if you're used to only caring about yourself, you're individualistic, being alone isn't necessarily the worst thing ever. But if you're someone who has community and interactivity within a group as the core of your identity, then isolation could be a very acute feeling of pain. And there's tons of research in progress looking at exactly those things. And it's one of those topics that's going to change fairly dramatically in the coming years. And then the last part of our feelings of pain is talking about how we actually don't feel pain. So the first of these is gate control theory. And I always give a little bit of a warning here. This is a strange theory, and it's described very vaguely in the text with intention. This is an old one. I think it was proposed back in the 60s, so it's a little bit weird to begin with. And they don't have a lot of hard evidence on how it specifically works, so we keep it vague for a reason. So you only have to know the vagueness of it. But the basic idea behind this theory is that incoming pain sensations, stuff that's coming up our spinal cord from those receptors, they have to pass through a gate in the spinal cord that could be closed to block off those ascending signals. And it's not a physical gate. It's not like something clamped down on our spinal cord. It's actually more of a theoretical gate, like a neuronal firing pattern that could disrupt those ascending signals. Um, that vagueness is why we don't go into more detail here. So that theoretical gate would probably be some kind of pattern within how the cells in our spinal cord work. And though they've had evidence for the outcome of why this one might be true, they haven't actually found a predictable pattern that lines up with what they'd expect. They don't actually have a mechanism that would drive this, even if they have lots of research that seems to be similar to what we should see if there were a game, if that makes sense. So we mention it because it's one of the big and first theories of pain. but we don't quite know how it might work or even if it does. So we have other theories that we could possibly have, these ones with a little bit more evidence and a little bit more believability, I guess. But we've already possibly mentioned endorphins just a little bit when talking about things like neurotransmitters. But endorphins are naturally occurring painkillers within the body. So where endorphins are produced or when they're produced, we tend to feel less pain. So they are found all throughout our central nervous system, and their presence seems to correlate with reduction in pain perception. And that's cool. We don't know all the specifics of it. We don't know all of those pathways, but it does seem to tie into our next idea, which is the descending neural pathway. So our endorphins seem to trigger a pathway that originates in the brain and moves downward to mediate or sort of start that suppression of pain. So instead of that gate where it cuts off or prevents the signal from passing through, this is where it tells other cells to inhibit or start suppressing our feelings of pain. So don't feel as much pain, don't fire as intensely. And so these endorphins start this pathway, and the pathway inhibits activity in the neurons that would pass along that signal of. And again, we don't have a ton of information on how all this works, but that one seems to have a bit of evidence, and we do see a reduction in firing for nerves or neurons that have been triggered by what they think is that pathway. All right. So at the very, very end here, it looks like we're really tight on time, but when you see how much is on these slides, it will work. We're going to talk about our kinesthetic and vestibular system. So our kinesthetic system just monitors the position of all the parts of our body. There are receptors in our joints and muscles. They register if our joints are sort of straight or bent. They measure if our muscles are tense or relaxed. So we know the position of our body because of all of these receptors. Signals from that kinesthetic system get transmitted to the brain along the same pathway as all that tactile stimulation. So pretty straightforward there. Our vestibular system is actually something that we already mentioned when talking about the ear. But our vestibular system responds to gravity, so the force downward on us, and lets us know our orientation in space. So if you remember those semicircular canals in our inner ear, those are fluid-filled canals. Gravity acts on those fluids, and as we move, it interprets how the fluid is moving as well. So standing upright, the fluid is down at the bottom. If I lay down, they'd be at the back. And when we move, we feel it slosh around. So if you've ever sat on an office chair, spun around really quickly and stopped, and you get dizzy, it's because the fluid is still sloshing. It still tells your brain that you're moving, but your eyes tell your brain that you're stationary. And the conflicting information makes you dizzy. But yeah, so those semicircular canals, they interact, or we can sort of perceive the behavior of the fluid within them to determine our orientation and our movement in space. So that's very critical for balance and orientation. If you've ever had an inner ear infection and it's caused issues with your balance, that's why. It's because it's messing with your ability to pick up on the location and the movement of that fluid. And as I said, very short on those two. So I always warn that it might be a great idea to know the difference between the vestibular and the kinesthetic system. So we do mention them very briefly, immediately adjacent to one another. So it might be a good plan to be able to tell them apart if a question were asked. But I will leave you there for today, and we will pick up and finish off this chapter, hopefully, on Wednesday with our visions